and my name is Riccardo Calandrelli from Dr. John Lab. And today I will present an overview about our study of endothelial dysfunction, where we try to understand what are the mechanisms behind the endothelial cell dysfunction using an integrative analysis, while mostly focusing on the RNA chromatin interactions. For those of you who are not familiar with endothelial cells, so what are these cells and where they are located? As you can see from this image, endothelial cells are the most inside monolayer of cells in the lumen of vessels, and you may not realize, but they are the largest tissue in our human body. Basically, they represent the direct interface between blood and tissue, and this direct contact with the blood flow makes them constantly subjected to any stimulus circulating in the blood. So also to all the detrimental factors that are present in blood in several diseases that can cause endothelial cell dysfunction. Now, diabetes is one of the most prominent risk factors, which affects 1 out of 3 people in the United States, and it can damage this layer. It does that by providing mainly 2 types of stresses. One is high glucose or hyperglycemia, and high glucose in the blood or hyperglycemia, and the other are several inflammatory factors. So our main goal with this study is to investigate how these factors lead to endothelial dysfunction, because of course, studying endothelial dysfunction would be significant to understand diabetes and its complication. Now, in order to achieve this goal, we established a UVEC in vitro system to mimic the temporal progression from healthy to diabetic states by using a combination of 2 stresses. As I said before, one high glucose, abbreviated with H to mimic hyperglycemia, and the other TNF-alpha, abbreviated with T, to mimic chronic inflammation. Then we generated 3 samples at 3 different disease stages. The first one, an healthy condition, or our control. Then another one, a pre-diabetic condition, where cells were treated with this combination of stresses for 3 days. And finally, a diabetic sample where the cells were treated with the same stresses, but for a longer period of time, 7 days. Then we used a combination of single-cell RNA sequencing in situ-IC and MRG to perform this integrative analysis I was talking about, and study and characterize this system from the transcriptome point of view, DNA-DNA interactome, and RNA chromatin interactome. So if we briefly look at the results from single-cell RNA-seq analysis, you can see how these 3 samples were well separated in the t-SNE plot and show these nicely separated 3 clusters. This means that our cells indeed underwent dramatic transcriptional changes during our treatments. However, if by looking at the PCA, we can see that we don't obtain these 3 separate clusters, even though there is already a path going from day 0 to day 3. This means that mainly these endothelial cells remain as the same population despite the difference in the transcriptome that we observed. But if we go further into the data, we can see how the single-cell analysis revealed the dynamics of the transcriptomic changes. For example, there are genes that are well-known markers of endothelial dysfunction, such as ICAM1, SERPIN1, FN1, and you can see how they are more expressed across single cells in the day 7 and day 3 samples compared to day 0. On the contrary, the NOS3, which is an hallmark of endothelial health, show the opposite pattern. The same results can be, or similar results can be observed from this table here, where we plotted a heat map with a list of the top differential expressed genes and grouped by functional pathways. And you can see how pathways associated with endothelial dysfunction, such as, for example, inflammatory immune response or extracellular matrix remodeling, etc., they show higher expression in day 7 compared to day 0. On the contrary, genes associated with the C function have the opposite trend. Interestingly, we also observed among the top differentially expressed genes a few long non-coding RNAs, which showed either increased or decreased expression in our dysfunctional cells. So taking together these results, basically we could confirm that our model indeed was reproducing the endothelial dysfunction and pro-inflammatory response that we were looking for. So moving on, we started with looking at very simple statistics from the genome interactome data. And first we asked whether the HT treatment induced any global changes in chromatin interactions from IC data. And here from this bar plot, we basically plotted the proportion of inter versus intra chromosomal interactions in our 3 samples. As you can see, there is not much of a difference from here. On the contrary, instead, you can see that IMARGI data showed a completely different trend. In this case, we observe a strong increase in the number of RNA-DNA interactions going from day 0 to day 7, which is, again, different than what we observe from IC data. Now, many of you are familiar with these IMARGI data since you are 4DN members, and we have presented this data several other times in other meetings. But for those of you who may be not, I will just show you here a quick example of what we mean by RNA-DNA interactions. So in here, you can see 2 regions, one on chromosome 2 around the link 607 gene, and one the chromosome over the chromosome 7 close to the SERPIN1 gene. And you can see how there are many more reads in the HT samples compared to control. But besides that, we have another piece of information, which is which read end is RNA and which read end is DNA, and as we colored the first in red and the second in green. Now, I've presented this specific example because I will go back to this a few slides later. But for now, I'm proceeding with an overview of how these IMARGI data look like. And as you can see here, they can be represented on a heat map, same as IC data. The only difference is that on the rows, we have RNA ends, on the columns, we have DNA ends. And this means that we don't have any more dimetric symmetry that we observed in IC. Now, if we superimpose over these metrics the location of superenhancers in the rows and in the columns represented by these blue lines, you can see that we can start to observe a non-trivial overlap, especially on the rows, namely RNA ends, of these superenhancers with hotspots of RNA-DNA interactions. Indeed, if we quantify this enrichment by comparing the amount of overlap of IMARGI read ends over the superenhancers compared to their genome average by length, we can see how both RNA end and DNA end in control and HT samples are way above this dotted line. And also this quantification reflects the fact that we seem to observe more enrichment and more overlap on the rows, namely RNA ends, than the column DNA end. But also, if we look at the superenhancer pairs, we can see how their trend, so when I say superenhancer pair, I mean all the IMARGI pairs where both ends are overlapping superenhancer regions, are located as superenhancer regions. And so, yeah, you can see the same trends as we observe for the global data. So decrease in intrachromosomal, increase in interchromosomal, but in this case, the amount of increase looks much more dramatic than the amount of increase in the interchromosomal looks much more high than the decrease in intrachromosomal. So all these observations made us wonder whether the changes on the gene expression in these functional endothelial cells could be caused by these interchromosomal RNA-DNA interactions between superenhancer, given this big change that we observed between the two samples. Now, to answer this question, what we did was to represent globally the superenhancer interactions using superenhancer networks like these ones. So in here, each of these blue nodes is a superenhancer, and edges represent interchromosomal interaction between superenhancers. But besides that, you can see that there are here nodes colored in different ways. So the red nodes are what we call superenhancer hubs, meaning those superenhancer establishing a very high, so establishing interactions with a very high number of other superenhancers that you can see. They're located at the core of the network, and likely they could be most important in the progression of the disease. But besides that, the networks also provided us another piece of information showed by the green nodes. So what are the green nodes? The green nodes are superenhancer hubs at the day 7 that were already present as nodes before, but not yet up. So this shows also a continuous expansion of superenhancer hubs from day 0, where we observe only one superenhancer hub, to day 3, where they go to 14, and finally day 7, where they go to 25. But here you also see several number of genes. So these genes are genes that are well known associated with endothelial dysfunction, and they overlap our superenhancer hubs. You can see that the only superenhancer hub in control was MALAT1, the MALAT1 superenhancer. And this was not surprising to us because we know that MALAT1 interact with a large amount of transcription active genomic regions in control or unperturbed cells. But if we move forward, you can see starting to appear SERPIN1, for example, THB S1 or BWF that are all elevated in diabetes, but also inhibitors of angiogenesis such as RANX1 and several other genes promoting EC dysfunction such as, for example, TRIO and XT1. So now I will show you a specific example of what a superenhancer interactions means, just for you to connect the information given in the network with the same information provided by the heat map. So here you can see again the same example I showed before of the region in chromosome 2 around the link 607 superenhancer, and the region in chromosome 7 around the SERPIN1 gene. So when I say link 607 superenhancer, is because both of these genes happen to overlap a superenhancer region. And you can see how, as the network showed, there are minimal interactions at day 0 between these two genes. Then these interactions start to increase at day 3, but become more prominent at day 7. Now, this is what we call interaction between two superenhancer, which is an edge of these networks. And now you can see how this edge that I have highlighted here in black was present in day 7, but wasn't present in day 3 and day 0 in our wet network. In parallel, also, I would like to show from IC data that there is, like we observed before, like we said before, no real difference in the interactions across the time points. So this would mean that we could hypothesize a model where interchromosomal RNA DNA interactions activate critical genes in the target genomic regions, and this would contribute to EC dysfunction. Now, the last step would be here to test this model. And what we did was to use this novel interaction established by the 7 between the link 607 and the SERPIN1. Now, this was for us particularly interesting because the importance or the relevance of the link 607 in endothelial dysfunction wasn't really much investigated before. And the SERPIN1 instead was one of the most important and well-known key regulator of EC dysfunction. Now, how we tested that? Basically, we disrupted this RNA chromatin interaction established by the 7. And how we did that? Basically, just by knocking down link 607. So when link 607 is knocked down, basically there's no RNAs establishing interchromosomal interactions with the SERPIN1. As you see from this graph, the knockdown not only reduce the level of the link 607 here, but also reduce the level of is let's call it paired SERPIN1 gene at day 7. So the importance of this observation is that the SERPIN1 activation is crucial to endothelial dysfunction. So this means that our hypothesis could indeed be being demonstrated, meaning link 607, so RNA is transcribed from link 607 on chromosome 2, reach further a region on chromosome 7 close to SERPIN1 gene. This activates these genes, and this activation results in several inflammatory responses and ultimately endothelial dysfunction. One last step was to reuse our single cells as an alternative approach for testing this emergent regulatory link between the link 607 and the SERPIN1. So again, very simply, we thought that if link 607 RNA promotes the transcription of SERPIN1 in dysfunctional cells, we should expect both of these genes to be co-expressed more in cells at day 7 or dysfunctional cells compared to cells in control. And to implement this idea, we can use this simple table, this simple contingency table, where on the rows we have the number of single cells showing co-expression of the two genes compared to those showing expression of link 607 only at day 7 and control. And a very high odds ratio indeed supports this hypothesis. But to go even further, what we can do could be to test this not only for SERPIN1, but also to all the other genes that showed an established interaction with link 607 at day 7. So what you see here is basically a subset of the network that I presented before, where we only take out the nodes that are interacting with link 607. You can see all of them were super-enhancer hubs. That's why here they're also plotted in red. And what we could do could be to perform the same exact analysis that we did for SERPIN1 for all the other genes overlapping the super-enhancer. So in total here we have 30 genes, sorry. And you can see that for each of these genes, we can have an odds ratio, as we did here, and we can plot this odds ratio in here in this violin plot. And actually, SERPIN1 was the top ranked genes showing association of co-expression with the dysfunctional cells. But out of these 30, actually, other 21 showed an odds ratio over 1, meaning the same property. And among them, there were RANX1 and Trio that, like we showed before, they are associated with endothelial dysfunction. Besides the in vitro samples, we also had some in vivo data, meaning in this case, we are doing, we did single cell RNA sequencing on actual donor vascular endothelial cells. So here we are comparing cells between diabetic and normal donors. Also, in this case, SERPIN1 was the top one ranked genes. RANX1 and Trio still appeared figured among the top 1 genes. And so this means that our in vitro model indeed recapitulates the in vivo data and the progression of diabetes. So to wrap up, since my time is almost over, we have shown how interchromosomal RNA-DNA interactions are increased in dysfunctional cells caused by diabetes, and also that indeed these interactions contributed to the activation of those genes that are responsible for the initiation of the cascades of events leading to endothelial dysfunction, such as the SERPIN1 gene that we have also further tested. And to support these points, we also show that these interactions that mediate the transcriptional activation have corresponding single-cell patterns. So the importance overall is that this specific work can reveal a mechanism that is not only significant, or may not be only significant for diabetes, but also through the progression of many other diseases. Here, we previously included this provocative COVID-19 because at the beginning of this pandemic, when this was starting to be studied, there were several published work that now even increased, where the COVID-19 was labeled as an endothelial disease, because most of these patients, especially the critical ones showing high blood pressure, thrombosis, or pulmonary embolism, suggested that this virus may indeed target the endothelium. To conclude, I would like to acknowledge all the NIH grants behind this work, and also I like that this project was carried out in collaboration with the lab of Professor Jen Bowman Chen at City of Hope. In particular, I would like to mention Lixia and Kim Jun, who were behind the majority of the biological experiments, and all the other colleagues at City of Hope and my lab. And you also may, if you're further interested in this work, you may check our paper that is linked here and was published last year about this work. Thank you for your attention.